Welcome, everyone. This is Kelly Eversole, the Executive Director of the International Wheat Genome Sequencing Consortium. And I'm joined today by Philippa Borrill, who is from University of Birmingham. And she's going to give us a webinar about decoding the wheat genome, or a polyploid wheat genome. But before we go into that, I'll give you a quick overview of the IWGSC, and we have more than 3,000 members. We are in more than 70 countries, or at least 70 countries, and we work with or have been engaged with over 800 institutes and companies, and we have 8 sponsors who have made our webinar series possible because of their support of our activities. So I want to thank our sponsors for their support. Our vision 2.0 is to really enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. And some of these activities include expanding our collaboration with Arbor Biosciences. We are literally in the process right now of publishing RefSec version 2.1, and that will be accompanied with our RefSec annotation version 2.1. It's gone to the publisher or to the journal this week, so we hope to have that out very soon and can release that for the entire community. We are still hoping to be able to get funding for at least 8 land races that can be sequenced at very high quality. And then we're obviously always looking for other elite wheat varieties and genomic resources that we can get academic scientists to provide through pre-publication releases under the Toronto Agreement. And then, of course, our webinar series. Our next webinar is actually going to be organized by the Phytobiomes Alliance, which is another organization in which I'm involved. But I think the next webinar that we're having there will be of great interest to many in the wheat community. It's about predictive biology, understanding and reversing the evolution of antibiotic resistance. So I think it'll be kind of an interesting twist on predictive biology and be very useful for many in the wheat community. So just to let everyone know, the webinar is being recorded and will be posted on the IWGSC YouTube channel in a few days. You can subscribe to the channel so that you never miss any of our presentations or any of our webinars. The presentation will be followed by a Q&A, so if you have questions, please submit those in the Q&A panel on your dashboard. You also have a chat panel. Don't use that for questions, but you are free to use that to reach out to talk with fellow attendees or even to send the organizers a question. Already, you can download both of our presentations, mine and Philippa's, if you go to the handout button, and that way you'll have those already and can follow along on the PDF at the same time if you'd like. So at this point, and without further ado, I'd like to turn it over to Philippa Borrill. She's a lecturer in plant biology at the University of Birmingham in the United Kingdom, and she's going to talk today about decoding the polyploid wheat genome using gene networks. Thank you for joining us and agreeing to do this, Philippa.
Hey, thank you, Kelly. Okay, so I hope you can all see my slides. And I'd like to thank very much the IWGSC for inviting me to give this seminar today. So today I'm going to be talking about decoding the polyploid wheat genome using gene networks. And I'd just like to start with a bit of an outline of my talk. And this is going to be, why is polyploidy relevant? Why is polyploidy relevant? Why now? Why do I want to talk about this topic now? And can we use gene networks to escape from polyploidy? So starting with why is polyploidy relevant? Polyploid plants are abundant in the world around us. So you can see on this slide several different examples of polyploid plants, and over 70% of flowering plants have a polyploid evolutionary history. And indeed, many of our major crops are polyploid. As you can see here, we have oilseed rape, cotton, potato, banana, sugarcane, and wheat, which is my favorite polyploid. And I think that now there are lots of opportunities using many of the latest resources that have been developed for wheat, including the genome sequence and many other resources that I'll talk about briefly today, that can allow us to start using wheat as a model system to try to understand polyploidy. And the information we learn about how genes are regulated in polyploid genomes using wheat as a model will hopefully be able to be transferred to many of these other polyploid species. So as a recap, wheat has evolved through the hybridization of several diploid ancestral species. The first hybridization event took place between the A genome progenitor Triticum auratu and the B genome progenitor, which is thought to be something similar to Aegilops baltoides, about 400,000 years ago to form a tetraploid wheat, wild Emma. And this tetraploid wheat underwent a second hybridization event with the D genome progenitor Aegilops tauschii about 10,000 years ago. And this hybridization event formed hexaploid wheat, which has three subgenomes, the A genome, the B genome, and the D genome. And I'll be referring to these quite a lot during this webinar. So these several steps of hybridizations mean that the wheat genome has three similar subgenomes, the A genome, B genome, and D genome. But not only this, the wheat genome itself is very large. So just to put it into scale, this is a representation of the size of the wheat genome, and these are some other common organisms. So Arabidopsis and rice were both very well known to plant researchers, and also the human genome. So the wheat genome is over 5 times larger than the human genome, which has really posed many problems in introducing genomics within research. But now we have really started to overcome these challenges with wheat, which opens up a whole range of new possibilities. So the wheat genome consists of 21 chromosomes, 7 chromosomes in the A genome, 7 in the B, and 7 in the D. And if we have a gene on chromosome 7A, for example, we are likely to find a gene copy on chromosome 7B and another copy on chromosome 7D. And these three copies of genes are known as homologs, and these are over 95% similar in the coding sequence, which raises many questions about their functions. Do these three copies of genes, the three homologs, have similar biological functions, or is it possible that after polyploidization, these three homologs have started to subfunctionalize or neofunctionalize? And that's something I'm very interested in and I'll talk about today. So one of the reasons I'm interested in this is because functional redundancy between these homologs does hide variation, particularly phenotypic variation. So in a diploid species, we would expect to see if we have a mutation in a specific gene in the homozygous state, if we mutate that gene, we would see a significant effect on the phenotype that it affects. And we can see this also in polyploids like wheat, for example, in dominant mutations like PPD2 and VERN1. We see that a single homolog is mutated, and this causes a very strong mutant effect. However, there are many genes in the wheat genome which have dosage effects. So, for example, we can see additive cases where if we, for example, knock out one of the three homologs, we only get a 33% effect compared to in a diploid species. Knock out 2 copies, we get a 66% effect. And we knock out 3 copies, we get 100% of the mutant effect that you would see in a diploid species. And examples of this include genes like GW2. So here we can see that as we increase the number of homologs which are mutated, we can see a significant increase in grain width, and we can also see an increase in 1,000 grain weight, increasing from 6% to 21% as we increase the number of homologs that are mutated. And this shows that we have this gradual additive effect, but just to note, this doesn't completely explain the full variation we see in diploid rice. So this might suggest that GW2 has slightly different functions in these two species, or we still don't fully understand its role. As well as additive effects, we can see non-additive effects between homologs. So for example, knocking out one copy might cause a small effect. Knocking out two copies might cause another small but slightly bigger effect, whereas knocking out all three copies will cause a very strong phenotypic effect. And in wheat, again, we see examples of this type of gene. These genes include GPC, grain protein content, and the waxy gene. And we also see cases of functional redundancy. This means that if we knock out one copy, the phenotype is exactly like wild type. We don't see any phenotypic effect. If we knock out two copies, we still see no phenotypic effect. It's only when all three homologs are mutated that we can see a phenotypic effect on the of the gene. And this is the case for genes such as starch branching enzyme 2 or the MLO locus. And really, at the moment, we don't know which genes within the wheat genome fall into each of these categories. However, we can say that many of the QTL that we've identified already in the wheat genome and identified the genes which underpin these QTL are likely to be in the left-hand two categories, so either dominant effects or genes that have an additive effect. So even mutating one or affecting one of the three homologs does cause a significant phenotypic effect. Yes, it's only 33% of what you would expect in a diploid, which makes QTL mapping quite challenging in wheat, but we have been able to detect these effects. But the consequence of this is that because we can only detect mutations or variation within homologs that fall within genes that have dominant or additive effects, we have a huge proportion of genes in the wheat genome that really potentially have hidden variation that we haven't been able to tap into in wheat breeding to be able to improve the wheat crop. And really this means that a large proportion of the wheat genome, we don't understand what these genes are doing or how they could be beneficial for wheat breeding. So the consequences of this is that many genes which have these non-additive or functionally redundant functions haven't been selected so far by breeders because it hasn't been possible to detect their phenotypic effects. So one example of this gene is GPC. So this is grain protein content, and it was cloned to be a NAC transcription factor called NAMB1. And I'll come back to this later as well. But in this gene, then the original cloning of this gene was through comparing an introgression line which had a functional copy of GPC, B1, compared to wild type genes, which wild type plants, which in wheat tend to have a non-functional copy. So actually the wild type already has one mutation. And you can see that the difference in senescence, which is how this gene was originally identified and through protein content, was that the difference in senescence was about 1.5 days, showing that just mutating one homolog has a very minimal effect on phenotype. But when you knock out two copies, you get a stronger effect between 5 to 7 days roughly. But when you knock out all three copies, you get a very significant effect on senescence, so approximately 25 days delay in senescence. And this is what we see here in this photo from the field, that wild type plants have already senesced, whereas the triple mutant plants are still completely green at the same developmental stage after anthesis. So this really illustrates the power that looking at just mutating one copy, we saw this only about 1.5 day delay in senescence, which also, as an aside, makes it very difficult to do genetic mapping because the phenotypic differences are so subtle, especially in a phenotype which can be strongly affected by environment. But by understanding the gene underpinning the GPC-QTL, so this NAC transcription factor, we could then go ahead and knock out all three homologs. And then you can see that this has a much stronger phenotypic effect and unlocks new variation that it wasn't possible to see by looking at, for example, different varieties of wheat. We can see these type of examples in other pathways as well. So these are two genes involved in starch synthesis, so the waxy gene and the starch branching enzyme 2A gene. And if we look at waxy first of all, then in wild type plants, if we set the amylose content to be a notional 100%, when one copy of waxy is knocked out, then you can see a decrease in amylose content. It's fairly subtle, but you do see a significant difference. When two copies of the waxy gene are knocked out, again, you see another decrease in amylose content, but it's still quite subtle. Whereas when you knock out three copies of the waxy gene, you see a very strong effect on amylose content with a near complete abolition of amylose within the grain. We can also see these really strong changes in phenotype with starch branching enzyme 2A. So a wild type plant, again, has 100% of amylose. When 2 copies of the starch branching enzyme 2A are knocked out, so 2 of the 3 homologs in any combination, you don't see any significant effect on the total amount of amylose within the grain. However, when all 3 homologs are knocked out, you can see this dramatic increase in amylose content, showing a dramatic change from wild type. And this really illustrates how this variation that it is possible to achieve in wheat phenotypes is masked by these copies, the homologs within the wheat genome. So coming back to my question, why is polyploidy relevant? Then polyploidy is common in many major crop species that we rely on around the world today. And yet polyploidy hides variation. So there's this functional redundancy within polyploid genomes between the homologs, which really limits the variation in terms of phenotype that we've been able to tap into. Coming to my second point, why now? Why can we start talking about this and really start making progress in this area? I think one of the reasons in wheat is that we now have a fully sequenced wheat genome. We assembled over 94% of the wheat genome, and we also importantly have a gene annotation. Kelly mentioned earlier there's work ongoing to improve this annotation. I think that's very important, that this isn't the final product. It's very difficult to get the perfect genome annotation. But I think that in this case, having over 110,000 for the wheat genome has really produced a step change in understanding gene function in wheat. And this genome sequence has also enabled the scaffolding of many different resources together and integrating different approaches within wheat, all using this common reference sequence. So for example, having a genome sequence means that now gene editing approaches can be accurately applied to knock out or alter the functions of genes in wheat. And this is the same for tilling mutants that can be used to knock out or alter the functions of genes in wheat as well. Beyond these methods to target gene function, there's also additional complementary information that can be obtained by using the wheat genome sequence. So for example, epigenetic approaches have been used in wheat, such as sequencing to look at DNA methylation, also approaches like mute-RenSeq to rapidly identify genes underpinning phenotypic traits. And close to my own research, we've been able to analyze RNA-seq data and also build regulatory networks using the wheat genome sequence as a tool to really help us with these analyses. And we recently published a paper which describes how all these different resources can come together, and many others that I unfortunately don't have time to mention today, to start understanding gene function in wheat specifically. And Christopher Wawi gave an IWGSC seminar a couple of months ago that's available on YouTube, talking about how these resources can be used to understand gene function. So if you want to know more details of this, I would highly recommend his seminar. Coming back to RNA-seq data, so now for wheat, there are over 850 RNA-seq samples that have been analyzed. And these range across an entire developmental time course. So here you can see a photo of some of the different tissues for which we gathered RNA-seq data. And this ranges from seedling emergence all the way through to adult plant and senescence. And this data is available through the wheat EFP browser that was built in conjunction with the EFP group. And as you can see, this has 70 different tissues and time points that are illustrated here, and they're in triplicate. So if you have a favorite gene and you want to know its expression pattern rapidly in wheat, this is a really useful resource for a snapshot of the gene's expression in one wheat variety. And also, there's another set of wheat expression data available on wheat-expression.com, which I'll just, hopefully the video will work. And you can see here that we can look at a gene's expression. We can sort according to the different ages of the plants. We can specify which age of plant we're most interested in. We can also resort according to tissue, and we can also compare the expression patterns between the 3 homologs. And this browser incorporates data from over 1,000 RNA-seq samples. So it's likely you'll find a sample that's relevant to your particular biological question. You can filter samples according to the tissue they come from, the age, whether plants have had abiotic or biotic stress conditions, and also what variety the samples came from. So it's a powerful resource to start understanding gene expression in wheat. And I'll come back to this later and how we've been using it in our senescence research. So coming back to my question about why now. So the publication of the wheat genome sequences really laid a foundation to build upon and understand wheat biology in the genomics era. And this has really helped the proliferation of a whole range of different functional genomic resources that can really help us to unpick what the functions of genes are within the wheat genome. Coming to my third part of my talk, can we use gene networks to escape polyploidy? So we're really interested in the field in improving a whole range of different traits in wheat, but we're really hampered by this functional redundancy, whereby if we use a conventional approach, for example, screening mutant populations, it might be there are many genes involved in the process that we just can't detect because the different homologs are redundant with each other. If you mutate one of the 3 homologs, you don't see a phenotypic effect. But in the field, we need to be able to improve wheat for a whole range of different traits. So breeders are interested in yield, quality, disease resistance, amongst a host of other traits. And we now have the tools to really start altering genes to improve phenotypes, to tailor them to the environment or growing end uses that we want to have our wheat grown for. And these include methods like gene editing, but also natural and induced variation that can be used within wheat breeding. But one of the main bottlenecks is how do you know which genes you want to edit or want to find variation in that will actually affect the trait that you're interested in? And to be able to do this, we need to be able to predict gene function. And today I'm going to argue that genomics can be one key way that we can start predicting gene function. So we can use information from pan-genome sequences from the over 7,000 RNA-seq samples that are publicly available now through the short read archive, and also I think there'll be increasing power of epigenetics to predict which genes will be important to target. So specifically today I'm going to talk about how RNA-seq data can be used to build a gene network, and then the gene network can tell us which genes or predict which genes will be most likely to influence our phenotype of interest. And then once we know which genes we're interested in for this specific phenotype, so here I've got an example of senescence, we can start using these techniques like gene editing or variation to then lead to hopefully crop improvement in the trait that we're interested in. In my lab, we're particularly interested in the process of senescence. So here we can see that over the course of the growing season, the plants turn from green through to brown. This happens in the summer in the UK. And this process of senescence is important because whilst the plant is senescing, this is also the period when the grain are being filled. And senescence helps to determine the balance between the yield and quality of the grain. And this is because the plant faces a choice during this time of its life. So the plant can either continue to photosynthesize, the leaves stay green. This allows more carbon to be fixed, which could potentially increase the yield, or the plant can make a decision to start to senesce. This will allow nutrients to be remobilized from the vegetative tissues like the leaves into the developing grain. And these nutrients could contribute to increasing grain quality, for example, by increasing protein content or increasing micronutrient content. And senescence is one of the mechanisms that helps to regulate this balance between yield and quality. But we don't know much about the genetic regulation of senescence. And we hypothesized that if we could understand this genetic regulation, we would be able to hopefully increase yields without incurring a yield, hopefully increase nutrient content without incurring a yield penalty, because often there's a negative correlation between yield and quality. So we know that genes regulate this process, but at the moment in wheat, we don't know that much about which genes are regulating this process. A couple of genes have been identified, but really this is largely a black box. And one way that you might think about identifying genes regulating a process is to do a forward genetic screen, so mutagenize a population and search for mutants which are affected in your trait of interest. And then you could use mapping to identify which genes are mutated. And we did do this in wheat. It is quite limited because you have these 3 homologs, so often a single mutation is functionally redundant with the homolog, so you can't detect it. And we also identified the NAMB1 gene, which underlay the GPC-QTL, which I mentioned earlier. So this didn't really tell us much more about the regulation of senescence in wheat, and we think this is largely due to functional redundancy between genes. If we knock out one copy, we just can't see any phenotypic effect, as you can see here. So instead, we decided to use a gene network approach. So by using gene regulatory network modeling, we thought we might be able to pick apart the effects of the 3 homologs. So we know we can analyze RNA-seq data in a homolog-specific pipeline. So we'll only be looking at expression of a specific homolog. And this would allow us to tease apart the effects of the 3 different homologs and hopefully identify novel genes which regulate senescence. So to do this, we looked at gene expression across time and then built a gene network. And within the network, we could identify genes that were highly connected, so hub genes. For example, in this toy example, then we can see the blue gene is the most highly connected gene in the network. It's connected to many other genes, and therefore it might be a good candidate to try to functionally characterize. We could then identify tilling mutants and test whether mutants in this gene affected senescence, and hopefully they would have a phenotypic effect. So we decided to look at a time course of senescence in the flag leaf. This is the upper leaf within the wheat plant, which provides the majority of photosynthase and nutrients to the developing grain. And we decided to look at a time course from 3 to 26 days after anthesis. And you might be wondering, why did we choose such an early time course of senescence? So even by the final time point, the leaf is still relatively green. And this is because the process of senescence isn't just the loss of green color, it's also a biochemical and transcriptional cascade that occurs before you can see any difference in leaf color. The other reason we wanted to look at the early events of senescence is because we wanted to identify the first regulators of senescence, because we hypothesized these would have the strongest effect on senescence. So we did RNA-seq on these 10 time points in triplicate. And to analyze our data, we tried using two different approaches. The first was co-expression, and the second was causal structure inference, which I'll focus on. So just to explain co-expression, we would have a gene with a specific expression pattern. So for example, the blue gene increases and decreases, and we could use this to identify other genes with a similar expression pattern. But the limitation of this approach is you need to know what expression pattern you think is going to be interesting, or which genes you want to find genes that are co-expressed with them. And because we didn't know much about the genes involved in senescence, we wanted to take a less biased approach to identify novel regulators of senescence. Therefore, we tried causal structure inference. Again, we have a gene's expression pattern. So this blue gene's expression increases and then decreases across time. We could then look at a second gene, this green gene, and it is also expressed in a similar pattern, but slightly later than the blue gene. And from this, the algorithm can infer that the blue gene is regulating the green gene. We could also have another gene, the light green gene, which has a similar expression pattern but inverted to the other green gene. And again, this means that we can infer the blue gene is regulating the green gene, although this would be actually a negative interaction. We could have an orange gene that increases in expression pattern but doesn't decrease, and this suggests that it doesn't belong in our network because its expression pattern can't be explained by the blue gene. And we could have a gene that's expressed before our original gene and go extending the network in all directions. One of the limitations with this approach is that you have to compare each gene against each other gene, so that means it's very computationally expensive. So we had to prioritize genes to include within our network. So we went from the 110,000 genes within the wheat genome using just the high confidence gene models. We focused on the genes which were expressed within our time course. So this gave about 52,000 genes that were expressed in the flag leaf from 3 to 26 days after anthesis. This was still far too many genes to be effectively run in this CSI algorithm. So we then decided to focus on genes that were differentially expressed during the time course, so genes that were upregulated or downregulated during this window of senescence. And this left us with 9,500 genes. This was unfortunately still too many genes. So we decided to focus on transcription factors, which gave us 341 transcription factors that were differentially expressed during this time course. And this was possible to run within the causal structure inference algorithm. And the reason we wanted to focus on transcription factors is because senescence involves major transcriptional reprogramming, and it's an active process where genes are deliberately turned on and turned off to bring about the process of senescence and nutrient remobilization. So we were hypothesized that transcription factors would be essential in initiating this whole process. So by understanding which transcription factors were involved, we would reveal master regulators of the process of senescence. And this is our result of our network. So you'll see there are 295 genes involved in this network. So we did lose about 50 genes that were transcription factors that were not involved in our network that we built. And just to explain this diagram, which I realize looks like a bit of a hairball, each gene is represented by a circle, and the lines in between genes represent connections between the genes, so that one gene is regulating the other gene that's connected by a line. And the size of the circles represents how many other genes each gene is connected to. So a larger circle means that gene is more highly connected. And we also can consider reading this diagram from the bottom to the top. So each of the lines actually has an arrowhead on it, but they're much too small to see, and it makes it very dense if I make them bigger. So you can imagine the direction of this network is going from the bottom to the top of the screen. So the first thing we wanted to look at was were any known genes that we know regulate senescence in wheat are found by this network approach. So on the left, I'm showing a picture of the NAM gene, where if you downregulate all the copies of the NAM gene, which you can see on the left-hand plant, you get a dramatic delay in senescence compared to wild-type plants. And this NAM gene is the gene that underlay the GPC-QTL that I talked about earlier in the context of non-additive gene expression and senescence. So we looked to see if these NAM genes were found in our network. And we did indeed find these NAM genes in our network. So we could find 4 copies of the NAM gene. So it's a little bit complicated. So the NAM1 copy, there are 2 paralogs, NAM1 and NAM2. And NAM1 has just an A and a D homolog within elite cultivars. The B copy is generally non-functional. And then we also found copies of the NAM2 gene, the B and the D homolog. So this really helped confirm that our network was identifying genes that were likely to be regulating senescence, and that gave us more confidence in this network. Also, interestingly, these genes are located quite towards the bottom of the network, suggesting that they might be some of the earlier regulators of this process. We then wanted to consider looking at new genes, and one thing we did notice was that there was another NAM2 copy, the A homolog, which we identified in our network. And this A2 copy had not been previously identified in work on senescence and this NAM set of genes, because it was actually the NAM A2 copy was missing from previous genome assemblies. So this made us think we should investigate what the NAM A2 copy is doing, whether it also has a role in senescence. And to do this, we used the tilling population. We crossed together single mutants in the A homolog and the B homolog. We decided to focus on tetraploid wheat to save time rather than working on hexaploid wheat, where we would need to look at all 3 homologs, because we hypothesized there might be functional redundancy between the homologs. So we crossed together a mutant in the A genome and the B genome and generated a segregating F2 population. We did a bit of back crossing to clean up the background as well. So looking at the phenotype of these plants, these are photos of the flag leaf during the process of senescence. And you can see on the left-hand side, these are all at the same stage, days after anthesis. And the wild-type plant, you can see on the left, has really started to senesce, whereas the double mutant plant on the right-hand side is still completely green, showing a delay in senescence. And just to put some numbers to the photo, you can see there's a delay in senescence of just over about 12 days in the double mutant compared to the wild type. We didn't see a significant effect in a single mutant of either the A homolog or the B homolog, indicating these might be somewhat redundant. We also looked at peduncle senescence. So peduncle is the part of the stem which connects the flag leaf to the ear, and we saw a dramatic change in peduncle senescence. So this was about the same as the flag leaf, so about 12 to 14 days difference in peduncle senescence. But in this case, we did see a significant delay in a single mutant of the A homolog compared to the wild type, which suggests that perhaps the A2 copy has a slightly stronger effect than the B2 copy, although the data isn't completely clear on this. So this showed that we could identify new genes that hadn't been previously identified using our network model, and we could pick apart the effects of different homologs and uncover genes that we hadn't been able to identify using conventional forward genetic approaches. So we never identified the NAM2 gene in our forward genetic approach. So this gene looked very promising, but it's very closely related to the NAM genes. You might think you could easily have predicted that that gene, if you found it in the genome sequence, would likely to be involved in senescence without going to all the effort of building a gene network. So of course we were interested in other genes in the network. So these genes that I've now highlighted in blue are all NAC transcription factors. And NAC transcription factors were of particular interest to us because they're known from a range of other plant species to be involved in regulating senescence. So we wanted to see whether any of these other NAC transcription factors within the network also had a phenotypic effect on senescence when knocked out. So I'm going to just show one example. So this is this NAC transcription factor here within our network. it's not right at the end of the network, but it doesn't have a particularly highly connected status and it isn't quite as early on in the network as the NAM genes. But nevertheless, we decided to look at this gene. And again, we found that there was a difference in senescence once we made our double mutant in a tetraploid background, as you can see in the photo on the left. And just to quantify this, you can see that we have a delay in senescence of the flag leaf, although this is not significant, which we think is because these plants have not been backcrossed yet, although that is work in progress. So we plan to rephenotype after 2 rounds of backcrossing to remove background EMS mutations, because these lines have quite a lot of background mutations. But we did see a significant difference in peduncle senescence in these lines in the double mutant compared to wild type, indicating that this new NAC does indeed affect senescence, and it could also be possible that it has a stronger effect on peduncle senescence than flag leaf senescence. So that's also a hypothesis we're going to test with our backcrossed lines. So using this approach, we've gone from having 2 previously identified genes regulating senescence in wheat to be able to rapidly identify new genes such as this new NAC transcription factor. And our next steps are really to start looking at how these differences in senescence relate to protein and micronutrient content, and whether these changes in senescence also alter nutrient remobilization. So coming back to our network, I've talked about a couple of genes out of the network, but there are still plenty of other genes in the network that look quite promising. So one that really stands out is this very large gene towards the bottom of the network that's very highly connected. And this looks like a very promising candidate gene. It's not a NAC transcription factor, but it is a gene we're going to be pursuing. And these are also other genes that look quite interesting because they're towards the very start of the network, suggesting they might be very key regulators in the process of senescence. So these are interesting candidate genes that we're following up by creating double mutants and characterizing their functions. But this network overall offers us a lot of information, but there are over 200 gene candidates. So how can we choose which to prioritize? So we could go just based on the network structure, but there is plenty of other information that could be useful to this decision process. So one resource we've been using a lot is the wheat expression atlas that we set up that includes over 1,000 RNA-seq samples. We've also used gene network information from NetMiner and ortholog information from Rice. So looking at the EFP browser for one of our candidate genes, what we did was look at the gene expression of this gene in flag leaves that were senescing. And this is an independent data set. It's from completely different variety. So we thought that if the gene was also upregulated in senescing tissues in this variety, that gives us more confidence that it will be involved in regulating senescence. So we prioritized candidate genes that were expressed over 2 times higher in senescing leaves than in other tissues in this independent data set. The second complementary data source we've used is the Gene3 network, which is available through NetMiner. And this network is a random forest produced network that predicts the target genes of transcription factors. So for example, we might have some particular target genes of a known transcription factor involved in senescence. So this is one of the NAM genes that I've mentioned earlier. And we could check if one of our candidate transcription factors also has similar target genes to that NAM gene. Is there any overlap? And this is built on an independent data set to our gene network about senescence, so it could add extra confidence to our candidate transcription factors. And there's recently been a paper published by Sophie Harrington from Christopher Wawi group in Norwich, where they looked at the Gene3 network and compared it to differentially expressed genes within knockout lines, and found that the Gene3 network is able to identify genes that seem to be downstream targets of specific transcription factors, increasing our confidence in the predictions made by this network. So using this information of expression data and the Gene3 network, we were able to find we prioritized 20 transcription factors which had additional evidence. And interestingly, the Rice orthologs, which we also used ortholog information to help us prioritize, and they were frequently involved in nitrate response and in drought responses. And this makes sense because we would expect that senescence may also involve nutrient remobilization, including nitrogen remobilization. This could perhaps be annotated as nitrate responses. And we also are not too surprised about the drought response link because it's known that in some environmental conditions, delaying senescence can improve drought tolerance. So this is an interesting angle to follow up in the future. So coming back to my question about can we use gene networks to escape polyploidy? I hope I've persuaded you today that we've been able to use RNA-seq data to build a gene network. And then by adding on complementary sources of information like RNA-seq data and other gene networks built from independent data sets, we've been able to identify candidate genes to follow up and functionally characterize. And this has enabled us to predict the function of genes involved in senescence. And this approach isn't just applicable to senescence, it could be applied to other time series developmental traits, and also to traits such as abiotic stress tolerance or disease tolerance. Any trait which you can look at a time series of gene expression could, in theory, be explored using this approach. And I think this approach is powerful because it allows us to really go past this problem of functional redundancy, where by mutating genes, we often can't see the effects on the phenotype due to functional redundancy. But by identifying the gene networks, we can identify the individual homologs and then prioritize genes for characterization using, for example, tilling mutants, where we can create complete knockouts by crossing together mutations in individual homologs, or by using CRISPR. So in conclusion, I asked 3 questions today. So why is polyploidy relevant? Why now? And can we use gene networks to escape polyploidy? So I think polyploidy is a major challenge, but also an opportunity for breeding crops such as wheat, because although it makes it more difficult, we also have this opportunity of huge amounts of variation that before we haven't been able to access. But now we can start accessing this. And this is largely due to the new resources we have available for wheat. So these are just a couple of examples, but there are many more resources that are coming online, which will really help us to exploit this polyploid genome. And I think that gene networks can be a powerful tool to help us unmask polyploidy and identify genes which we want to characterize in detail to affect particular traits. And I'd like to end by thanking people who've contributed to this work. So I'd like to thank the members of my lab at the University of Birmingham. We're a relatively new lab. We've been there for about 2 years now. And I'd also like to thank especially Christopher Wowie at the John Innes Centre and the members of his lab which have been involved, who have been involved in this work. Because a lot of the work that I've presented today was carried out whilst I was still at the John Innes Centre. I'd like to thank all my funders, and I'll be happy to take your questions.
Thank you very much, Philippa, for a great webinar. There are quite a few questions, and I don't know that we'll get to all of them, but I know that we'll try to do that. So the first question is redundance also observed at pathogen resistance loci?
Yes, I think so. So the MLO locus, then that seems to be functionally redundant between the 3 homologs. So you need to knock out all 3 copies to get a phenotypic difference.
All right.
And I think many disease resistance genes are present only as 1 homolog because a lot of them have come from introgressions from wild species. So it might not be universally true in disease resistance genes. So I think there will be individual differences, and also across the whole genome, we don't know how many genes fall into this functionally redundant category, or if they may actually have non-redundant functions.
So could you go into a little more detail as to what is dosage compensation?
Dosage compensation. So
you mentioned that early on in your talk.
Yeah, so dosage is really the amount of, well, I guess you could define it in different ways. So you could define it phenotypically, where you have the dosage expression of the gene, and then that contributes to the phenotype. So if you have 3 homologs which are all expressed, they could each contribute equally to the phenotype. You could get dosage compensation if you knock out one of the 3 homologs, and you might imagine a case where the other 2 homologs then increase their gene expression to compensate. So that might be dosage compensation.
All right. The RNA-seq data are from which wheat variety and the different platforms that you used for expression data?
So in wheat-expression.com, there's data from a whole range of different varieties. I think there's at least 10 different varieties. So the largest data sets are from Chinese Spring, which is the reference sequence variety. And there's also a large data set from Azania, which is a spring wheat. And that's the data that I showed in the EFP browser that has 70 different time points and developmental stages.
All right. Do you have any thoughts on how deletion lines, whole chromosome arms, might be useful, particularly for studying gene networks in light of some of the new resources?
It's a good question. I think they could be interesting, especially, I think one of the challenges of working with whole chromosome arm deletions is that you have such a strong effect on the whole genome because you've removed such a large proportion of genes that it's hard to tease apart whether you're seeing a primary effect of the particular gene you're interested in or whether it's a kind of whole genome scale rearrangement of gene networks due to that large change in the whole genome. So I think one approach that might be interesting is smaller deletion lines where you have just a smaller part of the chromosome missing, and that might be an easier way to dissect the effect on the gene network by removing like a very small part of each chromosome and then seeing what the effect of that is on the gene network. But it's a good question. I think there are lots of these genetic resources available in wheat, for example, the whole arm substitution or deletion lines that aren't really available in other species and gives us unique opportunities in the wheat community.
So what percentage of the wheat genes are expressed from the 3 subgenomes?
It's a good question. So we did some work on this that was published in 2018, looking at gene expression and balance between the 3 homologs. So I would say that about half the wheat genome is present with actually 3 homologs existing in the genome. So there's an A gene, a B gene, and the D gene. And of these genes, which is roughly 51,000 genes, then 70% of them are expressed at an equal level between the A, B, and D homologs, whereas about 30% have an unbalanced expression level. And it's quite rare that only one of the 3 homologs is highly expressed. So that's about 6% of those 50,000 genes expressed just from one of the 3 homologs.
Great. So is it possible to apply this method for detection of resistance genes or their networks?
I would think so, yes. So if you look at a time course of pathogen infection and disease progression and did RNA-seq on several different time points, so I think one of the one thing to say about the causal structure inference approach is you need to have multiple time points. So I think having fewer than 10 time points, maybe 8 time points would be okay, but you couldn't really use this approach on just say 3 time points. It wouldn't be enough to resolve the interactions. So you need quite reasonably high density time course information, but in principle, I think it could be used for other disease resistance networks. And I know Catherine Denby at the University of York has used this not on wheat, but I think on lettuce diseases and Arabidopsis diseases to understand the disease progression and genes involved in that interaction.
Do you know if anyone is actually pursuing that at this point in wheat?
I'm not aware of it, but I don't know.
Yeah, I haven't seen anything. So could you explain it in a little more detail how to develop double mutant populations that would enable you to study or enable one to study candidate genes?
Yeah, so all the tilling mutants are available through, well, you can check whether you have a mutation in your gene of interest using Plants Ensemble. So you can search for your gene ID, go to the Genome browser and check whether there are any mutations in your gene that you're interested in. You'd need to do this for each of the homologs. So for simplicity, let's talk about the tetraploid Kronos population, but there's also a hexaploid cadenza population where you'd need to also worry about the D genome. So you'd go to Ensemble Plants, check whether there's a mutation within your A homolog and the B homolog. We would usually try to choose a mutation that causes a premature stop codon because we think that would be more likely to knock out the gene and its function rather than choosing a missense mutation. Once you identify which of the lines has your mutation that you're interested in, you could then order those seeds from a seed repository. So there's one at the John Innes Center and also at UC Davis. Then you receive your seeds and then you would you'd grow up and cross together the A homolog mutant line with the B homolog mutant line. Select in the F1 using PCR-based markers for your seeds which had both mutations present, self those lines, and then you would have a segregating population to study your phenotype. Initially, you might want to backcross a couple of times to remove the background mutation load. I would recommend that anyone who's interested in doing this has a look at our eLife paper, Adamski et al., which was published this year. And also we have a wheat-training.com website, which has extensive details on how to work with wheat, how to grow the seeds, how to select tilling mutants, how to design the primers, really how to go step by step through the process. So hopefully that's a useful resource, which provides lots of information about that.
So in your estimate, how long does it take to actually go from the start? You've designed your experiment to actually having the double.
Yeah. I think a real game changer in this has also been speed breeding. So you grow wheat plants under 22 hours light. This has been work that's been pioneered largely by Lee Hickey in the University of Queensland. And by growing the plants under 22 hours light, you can halve the generation time. So with Kronos, it takes roughly 2 months to go from seed to seed. So if you need to do, say, 1 generation of crossing, 1 generation of selfing, and then maybe 2 back crosses, within about 8 to 10 months, you can have your plants ready to phenotype in a back cross state, which should be quite reliable. You can also do preliminary phenotyping before back crossing, or use multiple independent mutant lines to corroborate and kind of eliminate the effect of background mutations. So it's now much more feasible than it was before in wheat. So when I started this work, we didn't really use speed breeding, so it took much longer to develop the mutant lines.
So in your estimate, how much does it cost to actually build a gene network like what you've done?
I guess it depends on your sequencing depth that you want to do and how much your sequencing provider charges you per sample. But for our experiment, we needed 30 RNA-seq samples. So I guess people can estimate based on their sequencing provider, but say it costs £200, which I guess is what, $250 times by 30. This is really a test of my maths. Maybe someone else can do the maths, but roughly that amount. And then obviously the time for the bioinformatics analysis to be done, which is not super complicated, but it takes a while to interpret the data and prioritize your candidate genes according to your biological knowledge. It's not a magic bullet. You have to also do the groundwork to prioritize your candidates, because as I said, our network had over 200 genes. You can't, in wheat at this point, go through and characterize all of them, unfortunately. So you do still have to go through this prioritization step based on biological understanding of the process.
So how if you're a small company and you don't have a lot of bioinformatic resources, but you want to deploy this, is that feasible to do on your own, or is there a need to try to collaborate with an institute like yours to actually do some of the bioinformatic analysis and the heavy lifting in that regard? What makes the most sense for an SME that's trying to break into this arena?
I think that it really depends on the level of bioinformatics expertise in that institution. If there's no one who's ever analyzed any RNA-seq data before, then it's quite a steep learning curve, although there are lots of more easy to use platforms like Galaxy or Cyverse, which you can use to analyze the data. And actually the CSI algorithm that I built the network from, I used through Cyverse. So it's a point and click interface, which makes it a lot easier to get the software up and running. But to get to that point, you already need to have analyzed your RNA-seq data and have gene expression levels as count data or transcripts per million. So it's not, I would say it's a relatively easy to run bioinformatics pipeline, but it's unfortunately still not trivial if you don't have any background in bioinformatics. But if anyone's interested, I would consider a collaboration in the future on this topic. So get in touch.
All right, great. So one of the other questions was, as stay green is one of the target traits for heat stress tolerance, if senescence is related with GPC or QTLs per gene, can we target GPC genes or QTLs for heat drought stress tolerance? It's kind of a complicated question.
Yeah, that's an interesting question. So I've been thinking lately more about the connection between senescence and drought tolerance, and whether we can almost go the other way from the question, go from delayed senescence lines and test if they improve drought tolerance. But I think the idea of trying to improve heat tolerance through GPC could be interesting. I think we don't know enough about the mechanisms that regulate the grain protein content and how intimately that is connected with senescence, because I could imagine there might be some genes that affect grain protein content without affecting senescence and vice versa. But if you are looking at a key regulator like the NAM genes, then it's possible they might have a role in heat stress tolerance, although I haven't seen this reported in the literature.
Okay. Another question is, delayed senescence in wheat is getting much more time to get nutrition, and as a result, the grains will get more time to fully develop. But in a specific environment, for example, in terminal drought, are there advantages for the delayed senescence? I think you already kind of touched on that, in fact.
Yeah, so senescence is also highly environmentally dependent, and I'm sure that delaying senescence is not going to be beneficial in all environments. And in fact, if you're in a terminal drought environment, there's kind of a trade-off that you might want to try and delay senescence to extend the growing season, but it also might be a risk that you just really impact grain development because the grain might develop slower and then you might actually reduce yield. So it's really tailoring the process to be suitable to the environmental conditions in which the plants are growing. But I think it's interesting to now start thinking about the interactions between environmental control of senescence and also the genes that are involved in that process, although it's adding another layer of complexity to an already quite complex trait to understand, and we're really only starting to unpick the gene networks involved in that. But it will definitely be an interesting area.
So to make that a little bit further complex, is there any work undergoing that brings in the microbial networks that are occurring in the field, where you're actually bringing, trying to look at the interactions and the impact of the microbes on senescence or even drought tolerance?
That's a very good question, and I'm sorry, I don't know the answer. I think there are people. Sorry?
I have not seen anything really yet that's where it's tied in, but it would seem that maybe even adding that layer on top of what you've done already might be a very interesting study.
Yeah, that would definitely be interesting.
Yeah. All right, so getting to one of the next questions, is there isoform divergence between close homologs in expression?
Okay, so I would interpret isoform as different transcript variants and different expression patterns. We haven't specifically looked into isoform variation, so within the current annotation that's available for wheat, we think we are likely underestimating the number of isoforms between the number of transcripts per gene. So this analysis to date has been a little bit limited in wheat. But I could imagine it's possible that, well, for sure, between different homologs, you might have different transcript variants and gene structures. And for example, you might have an additional exon in one of the 3 homologs, or it might vary between different transcript isoforms. So I think it's quite likely there might be differences in the isoforms that are expressed between homologs, but at the moment we haven't done any systematic evaluation of the effect of isoforms on homolog expression, but it's an interesting area.
All right, so here's one last question. Can you speak on how causal structural inference was chosen for network analysis, whether DEGs were included with the transcriptome factors for the CSI algorithm, and how you view TFs versus target genes?
Yeah, so we chose the causal structure inference algorithm because it allows you to form these predictive relationships in terms of the directionality of the network, but also it's computationally lightweight enough that it could tackle the weak genome. So we did have to limit it down to these 341 transcription factors to be able to make the algorithm run. And that's really why we had to focus on transcription factors, because we went from the 110,000 genes, prioritized differentially expressed genes. So I guess this is DEGs from the question. And then from those about 50,000 differentially expressed genes, we then, 50,000 genes that were expressed in the flag leaf, we then looked at differentially expressed genes that changed in their expression pattern during our time course. That gave us about 9,500 genes. And then we prioritized the transcription factors from those differentially expressed genes. So all the genes in our network are transcription factors that are also differentially expressed during senescence. So it's possible there are some genes that are involved in regulating senescence that don't show differential expression during the time window we looked at, or that might show some sort of post-transcriptional modifications that will be involved in regulating this process. And I admit we're definitely blind to those genes and their regulatory properties on senescence. And I think that with these bioinformatic approaches, you have to sometimes make compromises in the number of genes you can include in your network, and also the value of the information you get out of it. So we also tried other techniques like WGCNA that builds a co-expression network to look at co-expressed genes. But as I mentioned, that's limited because you have to have an expectation of what your gene expression pattern should look like and which ones will be interesting. And then it's hard to prioritize which candidate genes to take forwards. But WGCNA does have the benefit that you can run it really with all the expressed genes. So you can build a WGCNA network with about 50,000 genes or even more. And we've done that for the 850 RNA-seq samples that are in the wheat expression browser, and that was published in 2018. So I would recommend having a look at that paper to see more about WGCNA co-expression networks.
Great. Well, that concludes our webinar today. There were a number of other questions, so we do hope to be able to provide answers to those subsequent to the webinar, and that will accompany the video, the recorded video that's on the YouTube channel. So again, I'd like to thank Philippa Borrill for being with us today, and hope we'll see you at our next webinar. And in the meantime, stay safe and healthy. Thank you again for joining us. Thank you, Philippa.
Okay, thank you. Bye.